All right, so today we're going to talk about DNA, RNA, how we make proteins, the cell cycle, and mitosis. So a lot of stuff here. It's only the last few slides of your PowerPoint though. But there's going to be a lot to really learn here. So that's why I wanted to separate this piece off. So this is where we're going to actually start in your PowerPoint. But I do actually want to show you just a nice abbreviated video that I found on YouTube, which I think is really, really great. It's called What is DNA? So hopefully it'll give us a little basis to go off and we can talk about some more of the things that they're talking about in this video.
And how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other, kind of like Legos, to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs, and organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine. AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process, as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood. I'm John Perry, and that's DNA.
Okay, so that gave us a little bit of a background or a summary of DNA, and we're going to talk about some of those pieces that he talked about briefly. So we talked about the structure of DNA. The structure of DNA is these kind of backbones that make up like the handles to the ladder, and that's made up of the sugar and the phosphate group. So then we have these nitrogenous bases. So there's 4 types: adenine, thymine, guanine, and cytosine. So those are the A, T, G, and C that you see. Now they can only kind of bond with those hydrogen bonds to each other, and there's a specific set there. So you can see that T bonds with A, G bonds with C, and so on and so forth. So that doesn't change, but what does change is the order of them up and down this ladder, and that's what makes everybody different. So it's important that we kind of understand those things. Not forgetting that DNA is really considered the cellular blueprint. It stores all of our genetic information, and it allows us to make tons and tons of proteins, which are the formation of life. We're also going to talk about, the DNA and the chromosomes when we talk about cell division. But now I want to show you the animation from Davis Plus, just to have a different spin on this and another way to kind of talk about it.
The DNA, deoxyribonucleic acid molecule, is one of the largest and most complex of all molecules. It is a polymer made up of many smaller molecules joined together in a sequence. The building blocks of DNA are millions of pairs of nucleotides. Each nucleotide consists of 1 sugar, 1 phosphate group, and one of 4 possible types of nitrogenous bases. The 4 types of bases are adenine, A, thymine, T, guanine, G, and cytosine, C. The phosphate group alternates with the sugar deoxyribose to form the backbone of the ladder. 2 different bases join together to form the rungs of the ladder. Base combinations follow a specific pattern. Adenine can only pair with thymine. Guanine can only pair with cytosine. The resulting ladder is twisted in the form of a double helix. A single strand of DNA would stretch about 2 inches if uncoiled. DNA is coiled to fit inside a microscopic nucleus. The sequence of bases is the genetic code. This code provides all the necessary information for building and maintaining an organism. Essentially, DNA serves as a cell's blueprint for life.
All right, guys, so let's take this one step further, and let's mention RNA, and then we're going to talk about how protein synthesis actually happens. So RNA, really its kind of job is just to copy the DNA and move that sequence of nitrogenous bases outside of the nucleus into the cytoplasm, where it can then be copied and where it can tell the ribosome what amino acids to bring together to form the protein. So there's transfer RNA and messenger RNA. So messenger RNA is going to go into the cell. That's those small strands the YouTube video was talking about, where it's going to copy the DNA after it unwinds. And then the messenger RNA can fit back outside of the nucleus. It goes out, meets the ribosome. The ribosome reads it in 3 sections. So it takes 3 of the bases, that combination of 3 bases together, and then it tells the transfer RNA to bring the specific amino acid. So the transfer RNA then brings that specific amino acid to the ribosome and deposits it there to form the actual protein. Okay, so we have transcription and translation. So transcription is when that messenger RNA actually goes in and reads kind of that DNA strand. Okay, and then translation is what happens then out at the ribosome, when the ribosome actually reads that strand and tells the transfer RNA to bring a specific amino acid to the ribosome to form a very specific protein. The video mentioned there's 20 different amino acids, so that's a ton of different combinations that you can make to form millions of different proteins. So it's important that we understand this difference between transcription and translation. So transcription is kind of the messenger RNA reading the DNA inside the nucleus. It then moves outside, meets up with into the cytoplasm of the cell, meets up with the ribosome. The ribosome then tells the transfer RNA molecule to bring a specific amino acid, and it deposits that amino acid there, and then we can make this amino acid chain, which will ultimately become the protein. So we really need to make sure that we completely understand the difference between those two things. Let's watch another animation, and just to reinforce some of that material.
One of the main functions of DNA is to provide information for building proteins. Proteins are the body's main structural molecules. They also contribute to almost every cellular function. The manufacture of proteins occurs in two main phases: transcription and translation. The transcription phase begins when the nucleus receives a chemical message to make a new protein. At that point, the segment of DNA with the relevant gene unwinds. An RNA enzyme then assembles RNA nucleotides that would be complementary to the exposed bases. The nucleotides attach to the exposed DNA and then bind to each other to form a strand of messenger RNA, mRNA. The length of messenger RNA actually consists of a series of 3 bases called triplets. Each triplet, called a codon, is the code for one amino acid. Once formed, the messenger RNA separates from the DNA molecule and moves through a nuclear pore and into the cytoplasm, where it begins the process of translation. In the cytoplasm, the messenger RNA attaches to a ribosome. The ribosome moves along the strand of messenger RNA reading the codons. Waiting in the cytoplasm are transfer RNA, tRNA molecules. Each transfer RNA consists of 3 bases, a triplet called an anticodon, that will perfectly complement a specific codon on the messenger RNA. Attached to the transfer RNA is the amino acid for that site, according to the genetic blueprint. The transfer RNA finds the 3 bases that are complementary to its own and deposits the amino acid. The ribosome then uses enzymes to attach the lengthening chain of amino acids together with peptide bonds. When each triplet has been filled with the correct amino acid and the peptide bonds have been formed, the protein is complete. The largest known human protein, Titin, consists of 33,000 amino acids.
So that just gives you a little insight into proteins, right? 33,000 amino acids. So think about how many different combinations of amino acids can come together to form proteins. And proteins really are a huge part of our body. All right, let's talk a little bit about the cell cycle and mitosis. So this cell cycle, you just need to know the different phases and what happens in each phase. So we have to have cells dividing. We need to replace worn out cells. We have to replace cells that have been injured. So the cell cycle encompasses the period from the beginning of one division until the beginning of the next. So it includes the following sequence of events. The first gap phase, the cell performs the tasks for which it was created while accumulating materials needed to replicate its DNA. So we are just kind of working on growing and getting the things that the cell needs. The synthesis phase, the cell makes or synthesizes an extra set of DNA. We need to have 2 sets of DNA, so when the cell division happens, each cell has 1 set of DNA. The second gap phase, the cell makes some final preparations for cell division, including synthesizing necessary enzymes. So kind of similarly to the first gap phase, it's not really actively dividing, it's just preparing, so it's getting the necessary components. And then the meiotic phase is actually where cell division occurs, which we'll talk about next. So the time between meiotic phases, which includes phases G1, S, and G2, is called the interphase because there's no cell division happening there. During the meiotic phase is actually where cell division happens. So after mitosis occurs, most cells repeat this cycle and divide again. We need cells to continually replicate themselves in order to keep our body working in the appropriate fashion. Some cells leave the cycle and enter a period of rest in which they don't divide. This can last for days, years, or even decades, but most commonly cells are kind of actively always working through the cell cycle and actively always dividing. So let's talk a little bit about mitosis, and that'll finish up the material. So mitosis is that kind of fourth phase of the cell cycle, and this is where cellular division is actually going to happen. So it starts out with one mother cell, we call it, because eventually it's going to end with two identical daughter cells. So it's going to go from one cell to two identical cells. So it's, cellular division or cellular replication. So we have multiple phases here. So in the prophase, we have chromatin. The chromatin begins to coil and condense to form chromosomes. Each chromosome consists of two strands, each of which contain a single molecule of DNA. The chromatids join together in the middle at a spot called the centromere, and we can see that here. So this is showing us the chromatids here. Where they're connected is called the centromere. During prophase, centrioles move to opposite poles of the cells and spindle fibers appear. We already talked about the centrioles before. The nuclear envelope dissolves and spindle fibers form in the cytoplasm. So we get this spindle fibers here. Here's our centrioles. In metaphase, some of the spindle fibers attach to one side of the chromosome. So then we get the chromosomes lining up along the center of the cell. In anaphase actually is where the centromeres divide, forming two chromosomes instead of a pair of attached chromatids. So this is where we actually get the chromosomes coming apart. The spindle fibers pull the newly formed chromosomes to opposite poles of the side in preparation for telophase, which is where actually the two nuclear envelopes come back together and we actually have two separate cells or one set of daughter cells. And each set has an identical set of chromosomes. It's not just half the number of chromosomes, it's the entire number of chromosomes that we need here. So when we talk later down the line on meiosis, which is reproduction and reproductive cells, it's a little bit different because we need to have only half the set of chromosomes. But here we're talking about each daughter cell having a full set of chromosomes. So let's watch one more animation here and then we'll be all set.
A key focus of a cell's life cycle is mitosis, when the cell splits into two identical daughter cells. Mitosis consists of the following four phases: prophase, metaphase, anaphase, and telophase. In prophase, chromatin begins to coil and condense to form chromosomes. Each chromosome consists of two strands, called chromatids. Each strand contains a single molecule of DNA. The two chromatids join together in the middle at a spot called the centromere. While the nuclear envelope dissolves, centrioles move to opposite poles of the cell and spindle fibers form in the cytoplasm. This is the final stage of prophase. During metaphase, some of the spindle fibers attach to one side of the chromosomes, and the chromosomes line up along the center of the cell. The next stage of mitosis is anaphase. The centromeres divide, forming two chromosomes instead of a pair of attached chromatids. The spindle fibers pull the newly formed chromosomes to opposite poles of the cell. Each pole will have an identical set of genes. Telophase is a reversal of prophase events. A new nuclear envelope develops around each set of daughter chromosomes. The spindle fibers disappear, and the cytoplasm divides to produce two identical daughter cells. The growth of organs and tissues in a developing child, the repair of damaged tissue after an injury, and the replacement of cells that die through the course of everyday living all involve mitosis. The only cells that don't divide through mitosis are sex cells.
So it's important that you understand these four phases and what happens during each phase, because that will definitely be a part of your test and it's something that you need to know going forward. So that finishes up this lecture.